Okay, my 232 class was canceled this morning about 7 minutes into it, I believe, with apparently some kind of bomb threat, and the campus was completely evacuated, and we all had to leave. So I came home, and I'm sitting here, and I'm thinking, you know what, we don't have a class period to waste if we're going to get through what we need to get through in 232. So therefore, I am going to go ahead and give this lecture. I actually backed up to the beginning of the blood chapter, leaving out the bloody sock pictures and things like that at the beginning. And I'll run through the lecture here, and you'll have it, and I'll post this so that you can all see it. But just know that I'm not going to lecture on this in class. So this is your shot right here. Let's talk about whole blood. Obviously, blood is pretty important. Blood is composed of two things. It's composed of plasma, which when you spin blood down, that goes to the top, and it's composed of the formed elements, which is all the cells, basically three types of cells: the red blood cells, white blood cells, and the platelets. Here's what it would look like if you took it from a sample of whole blood on the left there, and then spun it down. It would turn into this. The top part would be kind of pale yellow. It would be the plasma. The lower part would be the cells. 99.9% of those, just so you know, are red cells, but we do include the other things as well. The three formed elements, red cells, white cells, also called leukocytes. Red cells are called erythrocytes. The white cells play many functions. There's several different types of white cells. We'll go through them one by one later. And then the platelets are not really cells. They're fragments of cells that are involved in clotting, but they're very important. Hemopoiesis is a word you need to know. Hemopoiesis means the process of forming blood. And it happens in the bone marrow, in the myeloid stuff, remember? And it also has two different groups of cells, the myeloid cells and the lymphoid cells. We'll go through those later as we go through this as well. So, fractionation is a term for spinning blood down, basically. And when you spin it down, the whole blood, which is the stuff right out of your arm, gets separated, and then we can tell what it is, and we can test it. Three general characteristics of blood. It's about 38 degrees, so it's a little bit warmer than your body temperature is normally. The blood is thicker than water, so it has a high viscosity, and that viscosity is a term you need to understand. It just means thickness, so to speak. And it's also slightly alkaline. It's the pH of your body, which is really important. And just so you know, that pH is between 7.35 and 7.45. And in actuality, I mean, it's like right on 7.40 in almost every patient. Our blood volume overall is about 7% of our body weight. So if you weigh 100 kilograms like I do, which is more than average, then I would have about 7 liters, 7,000 cc's of blood in me, which would be about 14 pints here, which is, like I said, more than the average Joe because I'm a bigger guy. But please don't know any of those things. All you need to know is that it's about 70% of your body weight. And the reason that's important is because that's true of even babies. So if you have a 2-kilogram baby, you can judge just how much blood they have in them as well. 7% is a good number. Plasma makes up about 50 to 60% of the blood volume. Now here's where we get into all these numbers, and I'm going to give you one number to memorize, and then it'll be so much easier for you, I believe. So think of plasma as 55%. Obviously that's halfway between those two numbers right there. So the number we're going to think of is 55 which means that 45% is the formed elements, or the blood. More than 90% of plasma is actually just water. Remember that there's also extracellular fluid in your body, and plasma is an extracellular fluid. It's not contained within the cells. So the two major forms of extracellular fluid in our body are the interstitial fluid, the fluid between every cell in our body. Remember that every cell is surrounded by fluid, and the plasma, which is in the bloodstream. So what else does the plasma contain? Well, it contains water, ions, small solutes, and all of these things are freely exchanged between the plasma and the interstitial fluid, because they can go right through the capillary walls. The difference between plasma and interstitial fluid is basically the level of oxygen and carbon dioxide. Obviously, arterial blood coming straight from the lungs is very high in oxygen, and blood that's going back to the lungs is high in carbon dioxide, which is the waste product that we need to breathe out. There's a lot of proteins we'll talk about, and the thing you need to realize is that while the water and the small things go in and out through the wall of the capillaries, the plasma proteins do not. They're big, and they just don't go back and forth through the wall of the capillaries. Let's talk about plasma proteins for just a minute, because there are several types that you should be aware of. Probably the most common one is albumin, and I'm playing with my thingy here like usual, and I'm trying to make it work. There it is. Doesn't really show up there. So the albumins are about 60%. A lot of you have had egg whites before. That's basically albumin. It's a lot of proteins. I mean, these are proteins, and they involve fatty acids. They transport fatty acids. So the transport proteins in many ways are albumins. Notice that the globulins, which is about 35%, are also transport proteins, and so there's transport globulins as well. And they hook onto things, especially like hormones and specific lipoproteins and steroid binding proteins, things like that, to keep them in your bloodstream and traveling around for long periods of time. Fibrinogen is another protein that's found in your blood. Fibrinogen is a protein that forms clots. Now, it doesn't form clots until it converts itself into fibrin, and it turns itself into fibrin when it is exposed to factors that tell the body that, hey, we need to clot here to stop this bleeding, for example. So fibrinogen is the third of the major groups of plasma proteins. The serum is the upper part. If you take serum, if you take plasma, separate it off, and then let that clot so that all the fibrinogen turns into fibrin, then you can just kind of knock that fibrin out of it, and then you'll have what we call serum. So serum is plasma without the fibrin and fibrinogen. There's some other things that are dissolved in the plasma, which are basically the hormones and prohormones and enzymes, which are there in very, very tiny amounts, but yet they're very important because they have to travel around through the body to reach the target organs. Most all of these proteins that are floating in our blood are made in the liver, 90% it says there. There's also, outside of that, there's the antibodies, which are made by plasma cells. Now remember, antibodies are globulins. They're immune globulins. They're made by plasma cells. Peptide hormones is another set of proteins that are floating around in your blood. And remember, they're made by the endocrine organs. So the ovary makes estrogen, the testicle makes testosterone, the thyroid gland makes thyroid hormone, and so forth. Those are all proteins that circulate around in your plasma as well. Red blood cells make up 99.9% of the blood's formed elements. And so when you're looking at the lower part of that spun thing, my cat is about to walk across the picture here. Maybe not. You saw some whiskers, I think. So most of it is all red cells. And when you look at blood smears, which we'll be doing, you'll notice that 99.9% of all the cells are red cells. Red cells are really simple. They are basically just bags of hemoglobin. Hemoglobin is the red pigment that gives your whole blood its color. That's what makes it red. If you don't have enough hemoglobin, your blood is pale. If you have a really lack of hemoglobin, you are what we call anemic. We'll talk about that. Hemoglobin is the protein that causes us to carry oxygen. Obviously, hemoglobin binds onto the oxygen as it goes through the lungs. It carries the oxygen out to the periphery part of our body, like say our finger. That's the one I always use to illustrate with. Carries the oxygen out to the finger, and out in the finger, when the blood flows through the capillaries of our finger, it picks up carbon dioxide, the waste product, and then carries it back to the lungs where it can be expelled from our body. So hemoglobin is very important. That's basically what it does. You remember it from 112 because it was that wonderful protein that had 4 parts, and we learned about it, and we learned about sickle cell hemoglobin and so forth. We'll talk on those things again. Red blood cell count is the number of red cells in 1 microliter of whole blood. Now, what's a microliter? Obviously, it's a millionth of a liter. It's not very much. It's about a little bit of a drop, actually, not even a drop. And you notice that if you counted up all of the blood cells that are in that 1 microliter, it should be between 4.5 and 6.5. Well, for this course, think one number, and that is 5. So the normal red blood cell count is 5. That's 5 million per microliter, and you got to know the units on these things, because otherwise they don't make any sense. But this is simply counting the cells, and normal is about 5 million per 1 microliter of whole blood. Hematocrit is another number that we use a lot, often referred to as just the crit, also called the packed cell volume, and that's the percentage of the red cells when you centrifuge the blood down. So normally that's 45%. I've already mentioned that really because 45% is the formed elements at the bottom, but remember 99.9% of all those formed elements are red cells. Think 45%. 45% is a very normal number for both males and females. It's always higher in males than in females, and we'll talk some about blood doping and erythropoietin and performance enhancing drugs down this line here that will affect your hematocrit. The structure of red cells is really kind of cool. They are not round, they're not square, they're not cuboidal. They are they're called biconcave discs. In other words, there's a concave indent on either side, and the whole reason for that is to have a higher surface to volume ratio so that the oxygen can get in easier. The other thing about this shape is it allows these discs to form stacks called rouleaux. And that's really important as the blood cells are trying to get through these tiny, tiny little blood vessels out in the capillaries, say in the finger. They're actually, not only are they able to kind of line up as discs, but they can actually bend and flex in order to get through these. And once again, don't memorize these numbers, but just know that a 7.8 micrometer red cells can pass through a 4 millimeter capillary by folding itself up. So just know that red cells, their shape is very important, and their flexibility is also important. Here's a normal blood smear over here on the left. This is what they look like. Notice that they are, usually they are clear, almost clear in the middle, and they're darker red on the outside. Well, that's because of the shape of them over here. there's more hemoglobin between here and here than there is between here and here. So this middle part looks very pale. The outer edges look very dark. And one thing I've always used when I'm using my microscope to tell me, or to at least verify in my own mind that I'm looking at blood cells, is if you take the fine focus and just twirl it up and down and move it up and down just a tiny little bit, the cells will seem like they're winking at you over here. And that's because the clear area in the middle will become darker as you get in and out of focus. So keep that in mind. This is a scanning electron microscopy of red cells. It's basically 3D at a very, very high magnification rate. And you can see how they're biconcave discs. From either side, from the top and from the bottom, they are concave. This is what rouleaux look like. They're stacks of red cells working their way through these tiny little blood vessels. Remember that all little blood vessels, all capillaries, are lined by endothelium, which is a simple squamous epithelium. Here's a nucleus, and then, you know, all of this is just cytoplasm out here working its way down this little tiny tube, which we call a capillary. Red cells, blood cells, are very short-lived. Because they don't have a nucleus, because they don't have mitochondria, and because they don't have any ribosomes, they can't fix themselves at all. And they can't do any anaerobic metabolism. They live only about 120 days. So indeed, our red cells only last for about 120 days. Now, it's important that you realize that the red cells inside of your body live for about 120 days. A bag of donated blood that lives in the blood bank in refrigeration, it only lives for about 21 days. And we'll talk about that a little bit more later. But every day you are clearing out millions and millions of red cells that are getting too old. They're getting to that 120-day level, and then they get weeded out. We break down all the hemoglobin that's found in it and recycle it. So hemoglobin is obviously this big protein molecule that we've been talking about, and you can weigh the hemoglobin in a cell. And a normal hemoglobin level, once again, for adult male is more than for a female, but we're going to use the number 15 as the normal hemoglobin level. And the units on that are very important because hemoglobin is measured in grams. There's my cat again. Grams per deciliter. Now, a deciliter is how much? Remember, it's 1/10 of a liter. So therefore, it's 100 cc's. So in 100 cc's of blood, there should be about 15 grams of hemoglobin. And of course, 100 cc's of water would weigh 100 grams. Hemoglobin would weigh about 15 grams in that same volume. So that's another number to remember, is 15. The hemoglobin structure, you learned this back in 112, but basically it's a quaternary structure. In other words, it's made of 4 globular protein subunits. Each one of them has one molecule of heme, and each heme molecule has one iron ion. And that means that each one of those 4 globules can grab onto one molecule of oxygen. Now, think about that. That means that each molecule of hemoglobin can bind onto 4 molecules of oxygen. When the molecules are hooked to oxygen, we call it oxyhemoglobin. When the oxygen dissociates from the hemoglobin, we call it deoxyhemoglobin. For those of you who have ever been around trauma patients, you know that there are arterial bleeders. When a person has cut an artery, the blood that comes out of that artery will be bright red. The blood that comes out of veins, which is a lot more slow moving, will usually be dark purple. Now, the tricky thing is that once that dark purple blood lays there exposed to the air for a few seconds, it also turns quite bright red because it picks up the oxygen just from the atmosphere. But anyway, oxyhemoglobin, deoxyhemoglobin are the two major forms of hemoglobin that you need to be aware of. Here's a little picture. Here's a piece of here's a red cell, and down in it it's just chock full of these molecules of hemoglobin like this. Each molecule has 4 parts, 1, 2, 3, and 4. 2 alpha chains and 2 beta chains. Each of them has a heme molecule. This is the molecule of heme that's included in there. The heme includes an iron. And the 4 of them together can therefore, and each one of these heme molecules, this is what a heme molecule actually looks like. It's pretty complicated, and you don't have to know it, but just know that it has 1 iron in it. So there's a total of 4 molecules of iron in a single molecule of hemoglobin as well. So we've already said that the only function really that hemoglobin has is carrying oxygen, but it also carries carbon dioxide back. And so that's really another function it has. So it carries oxygen out to the periphery, out to the finger, and it brings carbon dioxide back. Carbon dioxide, when it hooks onto the hemoglobin, we call that structure carbaminohemoglobin. I love that word, carbaminohemoglobin. But then once it gets back to the lung, it disassociates from the carbon dioxide. The carbon dioxide is breathed out. You breathe in more oxygen. The oxygen takes its place, and it's carried out as oxyhemoglobin out to the periphery. The other thing you learned about in 112 that's important to think about is there's a small percentage of the population that has what's called sickle cell hemoglobin, or sickle cell disease, means they are homozygous for sickle cell hemoglobin. A sickle cell, the problem with it is that first of all, the hemoglobin is a little bit abnormal in it, and the reason that's a problem is that then when the sickled hemoglobin gets short on oxygen, gets a little bit desaturated, then it changes the whole shape of the cell. And so instead of being the nice biconcave disc like you see here on this side, it can shape itself like this. That's where the disease got its name, sickled cell. You also probably remember from 112 that the good thing about having a sickle cell trait, at least having a part of your hemoglobin being sickle, is that it protected you against malaria. So that's why the trait continues, because so many people live long enough to reproduce if they have a single sickle cell gene, whereas if they have both of them, it shortens their life, and they seldom make it to reproductive age, actually. So we've already said that red cells only live 120 days, so that means that about 1% of them are wearing out every day, which means about 3 million red cells per second. The numbers that we talk about here and the fact that our body has trillions of cells always kind of amazes me. But how do we get rid of 3 million red cells per second? Well, the way we do that is that we have these macrophages. We have lots of them that live in our liver and spleen, and a lot of them live in our bone marrow. And basically what those cells do is they run around looking for old, decrepit, worn-out red cells, and they eat them up. They simply eat them up, and they try to eat them up before the red cells really die and burst. We call that hemolyzing. Hemolysis is when the cells just bust for one reason or another and release their hemoglobin out into the plasma, which we don't really like to have happen. So what do we do with this hemoglobin? Well, that's important because we don't want to give it up. It's a protein. Our body has worked hard to build those proteins, and so we want to recycle it. And the globular proteins are converted all the way down into amino acids sometime. The heme, usually that's what happens to them. They're broken all the way down into amino acids, and then we recover the amino acids and we can build new proteins out of those. Remember, we only have 20 amino acids, 20 different ones. The heme is converted into biliverdin. Biliverdin and iron. Biliverdin is a protein breakdown product that's found in the plasma, and we'll talk about it a little bit more. Hemoglobinuria is something you don't really want to have happen to you. That means you're spilling hemoglobin in your urine. And the reason for that is that the cells are bursting and releasing the hemoglobin free out into the plasma. That's not a good deal. And when you pass it out in your urine, it makes your urine turn red. Now, just remember that that's different than the last term up there on the slide called hematuria. Hematuria means you've got blood in your urine. So this hematuria means you have whole red cells. When you look, and you can look at them under the microscope when you're looking at urine, and you'll see red cells in there. That's usually caused by a problem in your kidney or in your bladder. But nonetheless, hematuria is different from hemoglobinuria. Hemoglobinuria means that the cells have already burst, released the hemoglobin. Hemoglobin's in the plasma, and it flows out into the urine. The iron gets recycled. It gets picked up by transport proteins, carried back into the bloodstream. Usually picked up then by storage proteins. One of them is called ferritin, one of them is called hemosiderin. I just think you need to know that the iron gets picked back up, and we're going to recycle it. Now, how do we do that? Well, we do that by turning the biliverdin, which is that green product, it's the breakdown product of hemoglobin. It's converted into bilirubin. Now, bilirubin is yellowish green, and it's made in the liver, comes out in your bile. That's why your bile is yellow to green. And the bilirubin, as it says there, is excreted by your liver. If for some reason the system isn't working right, either you're not able to break the bilirubin down, or your body is hemolyzing its blood, for example. It's breaking down so much blood that you just can't break it down fast enough, then you get yellow. That's called jaundice. Jaundice is caused by a backup of bilirubin, basically, in your body. Now, normally, none of us are jaundiced. It's not uncommon for newborn babies to be jaundiced, to be yellow, because they are breaking down a lot of blood in the first week or two of their life. They're breaking down mama's blood and making their own. And so some of you may have had babies who had to be put under the bilirubin lights. It's a set of neon lights that helps break down the bilirubin in the skin so it doesn't build up too much in their brain. But anyway, not a very serious problem in general. That's what jaundice is, though. Jaundice means turning yellow. And if you've ever seen a patient that's jaundiced, you'll probably never forget it. The first thing that turns yellow generally is the whites of their eyes, their sclera. And it's a weird looking thing when you look at them and their eyes are yellow. That's the first thing you see. As the bilirubin level climbs higher and higher and higher, however, all of their skin turns yellow and it becomes very obvious. One thing about having too much bilirubin in your body is that it makes you itch. It makes your skin really itch like crazy, and these patients are miserable from having too much bilirubin in their skin. So just keep that in mind. That's another hint as to what's going on here. Now, once it's turned into bilirubin, normally we pass it down our bile duct, empty it into our intestine. It goes down and gets digested along with the rest of our food. Some of it is turned into what's called urobilin. Urobilin, you might get the idea that's from the urinary system, urobilin. And indeed, urobilin is reabsorbed out of the intestine, comes back into the bloodstream, and then it's filtered in our kidneys and passed out. this isn't all that important, but I think it is important to know that that's why our urine is generally yellow. Our urine is yellow because of urobilin, which is one of these breakdown products. Another group of these molecules are converted into what's called stercobilins. These are not reabsorbed out of the intestine, they stay in the intestine. And this is basically the chemical right here that makes our stools brown. So urine being yellow and stools being brown are both caused by breakdown of our own red cells and the reprocessing of those things. Here's a really good graph that I think you ought to study in detail, because I think it's important. If you start up here, here's a macrophage that's eating up a red cell, as illustrated right there. It's going to take that hemoglobin from that. It's going to turn some of it into amino acids that are going to go back to form other things. The iron is going to be moved over here and used in the bone marrow to make more red cells. These new red cells are going to be released and do their 120-day thing. About 10% of them are going to hemolyze. In other words, the cells are going to just break open, and that stuff is going to go, hemoglobin is going to be passed out here in the urine. That would be, if it occurs too much, that's when your urine really starts looking red, and that's hemolysis leading to hemoglobinuria. The rest of the bilirubin is turned into bilirubin over here, and it's manufactured in the liver. It's converted into bilirubin, which passes down through your bile duct into your intestine. In the intestine, it breaks down some of these bilirubin products, turns them back into urobilins, which are going to go here. Some of them are going to just stay in the stool and become stercobilins, as illustrated right here. So I think you need to understand this whole transformation of hemoglobin into bilirubin, into biliverdin, into urobilins, and into stercobilins, and how that all works, because hemoglobin is a very important thing for all of us. Let's talk about how red cells get made for a minute. They get made through a process called erythropoiesis. Erythro means red cell. Poiesis means manufacture. We only make red cells in one place, and that's in our myeloid tissue, our red bone marrow in adults. These stem cells, we have all of these stem cells that can turn into other things, but a lot of them mature to become red cells. The cells that are chosen to become red cells are called hemocytoblasts. That's kind of the first level. And these are stem cells that are found in the bone marrow that are going to start dividing, and they're going to become one of two cell lines. One cell line is called the myeloid stem cells, and myeloid cells are going to become either red cells. That's mainly what we're talking about here, is making red cells. But some of these myeloid stem cells are going to be manufactured and changed into some of the white cells that we use in our blood as well.